Hey guys, this is your genetics lecture on comparative genomics. I'm going to try to go pretty fast, but you guys can always pause it and go back and look at different spots. So comparative genomics, when you see that, you should think right away about, oh, this is about like conservation. How well are sequences conserved or not? It's a bioinformatics study of sequences and genomics and things like that. I'm both a good and a bad person to do this. So I am not an evolutionary geneticist, genomicist, or a comparative genomicist. I'm a neuroscientist by training, and I do a lot of functional genomics in the lab, which is something you guys will also talk about in this course. We also do a little bit of comparative genomics, and the good thing about that is we've had to kind of think through it and try to figure out the details of how it works. The good thing is hopefully you can think about it with me and kind of figure it out and understand the pieces of it rather than just all the complicated equations that a professional evolutionary genomicist would kind of understand. But the bad thing is that I don't understand all of the formal bits of it. Clearly, evolutionary genetics is its own entire course. So hopefully if you're interested, you can dive deeper into it. So the reason that I care about it comes from probably a reason that a lot of you guys are interested in genomics and genetics in the first place, which is trying to figure out answers to different diseases. So I have on the right kind of a standard workflow if you were doing whole genome, or in this case, whole exome sequencing. And so for us, we have patients that are seen in a neurology clinic that have terrible neurological diseases. And we want to know, it's clear that they're inherited, and we want to try to figure out what is causing their disease, right? What part of their genome is causing the disease? And so if you take a sample, you isolate the DNA, do target capture, run next generation sequencing, do base calling, and do variant calling, you're going to get a bunch of variants. And so for each individual patient, just looking at their exons, not right, their exome, not even their whole genome, there's about 50,000 exome variants. And these are only exome variants that NGS can detect. There's probably many, many more things like copy number variants that it's just difficult for next generation sequencing to really look at. But so 50,000. So which of those are the ones causing the disease and which are the ones that are just making this person different? Personally, I would like to do a functional assay to answer that question. But another approach that many people use is to try to impute which of those 50,000 are important using comparative genomics. So this is clearly one. But in addition to that, people use comparative genomics to try to figure out structure and function, interactions between different genes or even regions within a gene, how genes are regulated. You can use it in the synthetic biology realm to like redesign and build proteins. And of course, you can ask a lot of evolutionary questions like trajectories and evolutionary relationships between different things. Not untrivially, for virus-related research, viruses are quite hard to study, and one way that we can effectively study them is to use comparative genomics to ask how different strains of viruses are different. We can compare genomes of strains that are, our body's more tolerant to versus one that causes more inflammation. And so for the coronavirus, for example, there's a lot of comparative genomics studies that are trying to look into it. And it is a great way to just see what you can see. But generally, you're going to generate a hypothesis and not completely solve the question with comparative genomics. Okay, some of the big concepts. We're going to talk a little bit about evolution, talk to you more about conservation, and then a little bit of synteny, and then we'll break the rest of it up into more videos. Okay, evolution. Since I'm not here in person to actually call on people and ask you the question, hopefully you can pause it and try to answer the question, what is the definition of evolution? Hopefully you can answer it and you remember that it's the change in allele frequency over time. And so let's break that down a little bit because we're going to need some of this to actually think about how conservation and just in general how we can use comparative genomics. So I've drawn for you here 4 different sequences, so just 14 bases. And you can see that there's, let's pretend this is on the Y chromosome of 4 different males. Right, so we have 4 different males. there's only one copy of the gene because it's the Y chromosome. And you can see that in this population, there's just 4 alleles, right? Sorry, there's 4 sequences, there's 2 alleles. There's the C allele and there's the G allele. So at this site, there's just 2 alleles in the population, and each are represented at 50%. So this has nothing to do with evolution, it's just a snapshot, right? Snapshot in time, and you can see like, oh yeah, there's 2 alleles in there. They're represented in the population at 50%. So if I now showed you, either a time point before this or after this, and this frequency change, right, there's either more of the C allele or more of the G allele, then that would be indicative of evolution. Now remember that just because it's evolution doesn't mean there was any selection or anything else. There's could just be random, right? This could just be noise. It's still evolution, right? Because evolution is simply the change in allele frequency over time. So what would this be if this changed? There's a few options there's a lot of different options if you study evolutionary genetics, but the options that we care about are, there's basically two options for selection. And the simplest and the one we'll focus most on is negative selection, which is also called purifying selection, which means that if one of these alleles gives you a fitness advantage, or conversely, other alleles, it's mostly if a particular allele gives you a fitness advantage, then that allele will be selected for, and it will end out becoming more common in the population, right? So it's called negative selection, and you've purified for a particular sequence. And we'll see what that means in terms of like mutation rates. But you can imagine that means that the mutation rate in this region is going to be lower because you're trying to keep this specific really good sequence. But you can also have positive selection, and that means that this region is going through a lot of change. So there's changing this region is helpful, and so you see actually more changes, more different kinds of alleles in this region than you'd expect by chance. So, and speaking of chance, that's the background. So there's changes happening everywhere, but they're not because of selection. You can think of it as just like background noise. So this is just background mutation rate and a lot of other things that are going on, like drift. But so we call this neutral evolution. So it is still evolution, it's still changing the allele frequency, but it's just at a background neutral rate. So this is it. This is kind of like the key for comparative genetics, is understanding that if we can find a background, which is how we get this neutral rate, then we just ask, oh, okay, are things mutating faster than the neutral? If so, that's positive selection. Or are they more conserved than the background, less mutation, and that's purifying selection or negative selection. So make sure you write at least that much down. Write the definition of evolution, write the two kinds of selection, and then make a note that most of this is trying to figure out what is the signal, right, positive or negative, over some background. So how are we going to figure out the background? For coding regions, it's fairly simple because we can take advantage of the fact that some mutations change the protein code and others do not. So basically synonymous versus non-synonymous. And the classic way to do this is using something called Ka over Ks, which is just the non-synonymous rate. In other words, if you get a mutation that actually changes the amino acid, that's the non-synonymous rate, versus the synonymous rate, right? How many mutations are going on in a coding region that don't change that. And it's because you guys remember things like the third base pair wobble. So there's a lot of, you can see kind of the background mutation in the synonymous rate, but then part of the non-synonymous rate is considered some kind of selection. So in this case, a large Ka over S greater than 1 means that area is under positive selection, right? You're seeing more changes there. The rate of change is higher than you'd expect in the background. So positive selection. And if you saw something less than 1, it means that this area is, whatever region you're looking at right now, is undergoing purifying selection, right? So it's being kind of locked in, and therefore you would see conservation of that sequence. So coding is pretty straightforward. You won't have to actually like calculate any of these things. That is not the goal, but just to, if you see these words, K over KS and negative selection, and just to be able to think about it. For non-coding regions, it's a little tricky, right? Because how do you figure out what is the neutral rate in a non-coding region? There's not such obvious rules. So there is regions of the non-coding genome that we understand, like regulatory regions. And then there's other regions that people used to think were junk, but very likely they are not just junk. And so where do we look to get the background? It's a little more complicated. There's many papers that come up with many different strategies. I'll tell you about 1 or 2 very simple ones. But the idea is generally the same. Okay, so conservation is an example of purifying selection. That's what you see. But here, what we're talking about is sequence conservation. And it's just the question of, well, how conserved is a region? Now, conservation and like percent alignment or quality of alignment are closely related, right? So when you guys run a multi-sequence alignment or any kind of alignment, you can get a score, right? Which is like, oh, how well did my sequences align? How similar? What's the percent similarity between these 2 sequences? So I should have written this on the slide, but what is the difference between percent conservation and percent similarity? They're definitely very closely related. So any set of sequences that you can align, you can produce like a similarity score, like how similar are the sequences. But that same number it's related to conservation, right? Conservation has 2 other pieces to it. One is the evolutionary piece that I just looked at. You're always going to try to compare it to some background rate, because you want to see conservation compared to some background. And then the other thing is the things that you're going to compare in conservation are going to be, for example, different species, usually is going to be the case. Whereas for sequence similarity, it could be anything. Anyway, pretty simple. So to look at conservation, we need some species to look at. We need to look at some sequence amongst different species. It could be different individuals, but it's usually different species. And we're going to do a multi-species alignment, which is a form of multi-sequence alignment. So how do you choose the species that you're interested in? Obviously, whatever is your host organism, you want to make sure to look at that. But which other species should you choose? It's going to be a balance between choosing closely related species and distantly related species. And here's what the balancing act is about. Because with closely related species, the good thing is that your sequences align really well. But the bad thing is you don't see many alleles, right? There's not many mutations in closely related species. So you don't get a lot of data. So you can see this here. This phylogenetic tree has lots of different species of yeast. And just to show you how different Cerevisiae from Paradoxus is, it's about the same differences from a human to a monkey. And you can see that these various species of yeast have diverged over hundreds of millions of years. And so they are as different from each other as we are from, say, fish. So a huge diversity here and many millions of years of evolution. But so anyway, if we just compare 2 closely related species, you can see here in this 2-sequence alignment that almost everything is yellow, and in this case, yellow means identical. So not a big surprise, closely related species have mostly identical sequence, and the regions that aren't identical are fairly sparse, so we can't get a great picture of overall conservation. On the other hand, if we use distantly related species, the problem is it's really hard to align the sequences because there's too much difference. So if you can't align the sequence, you can't ask what is the similarity between different positions. But the good thing is that you do see a lot of changes. So the answer, of course, is perhaps straightforward, is that you need to use a mix of some species that are closely related and then kind of add in links that get you to more and more distantly related species. Having the links in there will let everything align, and having the more distant species will help you get sequence divergence, which helps you get a better measure of conservation. Okay, so let's throw a little bit of numbers into this, but not too many. So here we go. We're going to do a sequence. We're going to ask about the conservation between human and mouse of this particular region of interest. And so you can see there's 12 bases here, or 12 aligned columns, and 11 of those are identities. When you're doing nucleotide sequence comparison, there is a lookup matrix of, what is C to T versus C to G, but really you can just think of it as 1 or 0. Is it identical or not? When you're doing amino acid, there's the BLOSSOM matrix that you can use to say like, oh, well, these two nonpolars are fairly similar to each other, so that's going to be not so different as when you add a polar thing to a nonpolar amino acid. So there's different ways you can score. But for here, we're just going to do, it's nucleotide, we're going to consider it binary. So in this case, we have 11 identities, so this would get a score of like 11 out of 12. Okay, so if we want to measure conservation, I already told you guys we're going to try to look at this over the background. So here's two, let's say, randomly chosen background regions. You can see the first one has 10 identities, and the second one has 8 identities. So if you guys were inventing a conservation score for the first time, you'd probably say, well, let's take your region of interest and then show its value compared to, or like subtracted from or divided by the average of the background region. So conservation scores are approximately equal to this. There might be some other factors that are thrown in there, but approximately they're like, okay, we've got 11 alignments here. And then we'll take the average of similarly sized regions, right? All these regions have 12, so the denominator doesn't really matter. Right, so we'll average 8 and 10 together. So 11 minus 9 equals positive 2, meaning that our region of interest has 2 more bases than you'd expect from neutral evolution. So assuming that the background regions are under neutral evolution, these are just random background changes, the region of interest is more identical, so this is purifying selection, so you can say the conservation score is +2. And maybe you divide that by 12 and you'd say it's, 2/12 per base is the conservation score. Pretty simple. So a paper back in 2002 asked, well, if we compare human and mouse, can we figure out how much of the genome is neutral and how much is under selection and how much selection is under, how conserved is it or unconserved? And so what they did is they tried to align the entire human and mouse genomes with the reference sequences they had then. And they did this in 50 base pair chunks, and they were able to get 14 million aligned 50 base pair chunks or regions. So it's really just like I showed you a second ago, except instead of 12 base pairs, it's 50 base pairs. And so here's what they saw. So forget for a moment that it says conservation score in the bottom. And let's just say this is the number across all those 14 million sites. On the x-axis, this is the number of identities. And on the y-axis, this is the number of regions that had X number of identities. Right, so it's just a histogram. And without knowing anything else, you'd say, well, you know what? If we assume that most of the genome is under neutral selection, right, is not actively under selection between human and mouse, then this mode, this value right here, would tell us the background mutation rate. And that's what this paper did, and that's why they have conservation score here, because then they just subtracted everything by that number, and so this becomes 0. And so this is somewhere in this red region is the, if I was drawing it, I probably would draw, like the end of it here and here. But so you'd say this is the region under neutral evolution. Here's the region that's negatively selected. There's some region over here that's positively selected. And what they came up with after their calculations was that if you take into account that only about a quarter of all of the genomes aligned, and they put into this analysis, that about 5% of the human genome is under some form of selection, which is, in a way, a pretty big number, right? It's millions of base pairs if it's 5%. Okay. So who cares? How do we use this? I'll show you a few different ways, but one way, just to come right back to where we started. Remember, initially I said, if you get these patients with a terrible neuropathic disease into the clinic, you think it's genetic, you'd like to know what gene or what variants are causing it. How can we go from 50,000 variants down to fewer variants that are interesting? And one way to do that is using conservation. So the assumption is fairly straightforward, that if an area is very highly conserved, right, or is being negatively selected, that messing with that area is probably going to damage the organism, right? So if evolution has been selecting for a particular allele, but you see a variant in that region, one could hypothesize that that variant could be dangerous or pathogenic. And many different fields of biology right now are trying to assemble a lookup table, and you guys will hear more about this when you talk about massively parallel technologies. But the lookup table is basically to try to figure out, okay, variant, on chromosome 3, position 50,276, an A to a G there. We want to know if somebody comes into a clinic that has that specific variant, is that pathogenic, or is it benign, or is it epistatic with other things? We want to know something about it. So building a lookup table where we know something about certain variants is one of the major goals of precision medicine, I guess. Anyway, here's how you can use conservation to try to do that. By the way, I am actually against assumptions. So I think it's a reasonable hypothesis that putting a variant in a conserved region is going to be pathogenic. But I think that there's an incredible number of exceptions. So using something like this to filter out variants is potentially dangerous to the field. I think we're going to have to do functional tests for all these. But this still works, and I'll show you how well it works. So this is a relatively new paper that's using conservation scores to try to predict whether a change in a particular spot is pathogenic or benign. And they're doing this by something called a shared taxa score and local identity. So the shared taxa part is the slightly more complicated part. So if you look at a tree here, this is like a simplified tree. Shared taxa has to do with like how many shared edges of the tree do you have between humans and rats, or humans and fish, or cellular organisms, right? So the closer you are, like humans to humans, have a lot of shared taxa, and then it goes down. And then local identity, which is this column, just says like, okay, so at this level, right? So at the level of Homo sapiens, here's our reference sequence, here's what we're trying to query. And you can see that, the shared taxa is 31, it's just a lookup table. The local identity is 7, 1, 2, 3, 4, 5, 6, 7, right? There's 7 identities here. And as you go down this tree to organisms that are further and further away, or basically have less and less shared taxa, you can see that the local identity generally drops. And they use a combination of the shared taxa and local identity to build what they're calling a variant shared taxa score. It works fairly well. So they did this against a benchmark set of variants that are already known in ClinVar to be pathogenic, aka deleterious, or benign. And what you can see is that high VST scores tend to go along with benign, and low VST scores tend to go along with deleterious. In other words, a low VST score says that it's very conserved, and a high score says it's less conserved. So it's clearly there's information here, but you can see that there's a lot of overlap and a lot of fuzzy space. So this is good. We need to understand this, but it's not quite enough, of course, to call variants. So in addition to coding regions, which are pretty well conserved, there's other regions of our non-coding genome that are like really well conserved. So between human, mouse, and rat, there's about 500 sequences that are greater than 200 base pairs long, which are completely identical, right? So the completely identical sequences that are longer than 200 base pairs, there's about 500 of them. And fully identical sequences that are more than about 100 base pairs long, there's about 5,000 of those. So there's regions, let's say 5,000 regions that are at least 100 base pairs long and are like perfectly identical between these three species. When you guys look at a genome browser, you would see something like this. There's often this conservation track. Many of the conservation tracks come from something called the Maltese alignment, M-U-L-T-I-Z. And if you expand that, there's different varieties of it that have different number of species or organisms that are in there. And it's a conservation score like we talked about above. And it shows these regions that are very highly conserved amongst a variety of species. So, for example, this one might be one of these, an example like one of these 500 sequences. So what are these? If they're not part of a coding region, what are they? You can see it. This is this region here, for example. And here's a coding sequence. Here's a little bit of coding sequence. But then this upstream region is really well conserved, but there's no coding sequence. I think you guys already know the answer. This is some kind of a regulatory sequence, like a promoter or an enhancer. And so this is a great way for a gene that you guys are, maybe you're studying a gene that is that is very interesting to your lab for one reason or another, and not a whole lot is known about it. You can use conservation to look for regions within the gene that are likely to be of regulatory importance and just to find out how well conserved they are or not. And of course, you can do this down to the single nucleotide level. You can do it to the level of a codon, whatever you like. But so that information is all available, and now you know a little bit more about what it means. Okay, the last bit that I want to tell you about in this video is synteny. So conservation is trying to compare within an aligned column, how frequently is the base the same, or how often does it change, right? So what's the percent similarity across an aligned column? But that's not the only place we can look for similarity. The other place is the ordering of some unit within the genome, right? How are they, what's first, what's second, what's third? And that order may be conserved even if the actual bases are not. So this is the concept of synteny. So I'm going to show you two examples of this. The first one is showing you the location of genes on a bunch of different genomes. So you can tell that each row here is a different genome. So on the very top is a cow genome, and then a goat genome, and then a sheep genome, et cetera. And at the very bottom, below the break, are some bird genomes. And this is done in reference to a gene called SARM, which you can see in the middle here. And what it's showing you is that it's not showing you the actual genomic position, but it's showing you the ordering of these genes on the genome, 5' to 3'. So first comes POLDIP2, and then TMEM, and then VTN, and then SARM, and then this SLC, FOXN, and UNC119. And you can see that that series, whether or not their sequences are conserved, the order of those genes is pretty well conserved amongst the variety, pretty large variety of mammals and also of birds. So this is pretty strong synthetic conservation, or it's just called high synteny. Okay, so synteny is just the preservation of an order of, it could be anything. It could be domains, it could be bases. In this case, it's genes. But that's what synteny is. And so you can see that also between mammals and birds, there's a syntenic unit that starts with this pol dip2 and continues way down to whatever this pink gene is that I didn't label. And you can see that, that is maintained between mammals and birds. In mammals, there's another syntenic unit that starts with the green and goes this way, but that's different. It's not conserved in birds. There's something different going on here in birds. So we don't 100% understand what this means, but again, the assumption would be that genes that tend to be in syntenic units are probably together for a reason, and whereas genes that are frequently found to jump around or to be moved around in different genomes, it's probably less important for them to be together. For example, they're regulated in a different way. And this is all, synteny was a concept that was around before the 3D genome was discovered. And so there's probably a similar version of synteny for 3D genomes, but that is not my area of expertise. But the concept is still the same, right? Is there some structure that's preserved across different species or across evolution? And if you're within that conserved structure, it tells you something. It's not always clear what, but it tells you something. So here's a paper that just came out 2 years ago showing that in mammals, that 87.5% of genes that we have are in just one syntenic cluster amongst a set of like 100 different mammals. In other words, if you pick a gene at random, you're probably going to pick one of this 90%, where across all 100 mammals that they had in the study, that gene always has the same set of neighbors. In other words, it's in just one cluster. Only 10% are in two different clusters, meaning that in some mammals, they're with genes X, Y, and Z, and in other mammals, they're with genes A, B, and C. Right, so they're like rebel genes. In some genomes, they're together, and in other genomes, they're with a different set of friends. And this picture is showing you examples of these genes that are in two clusters. And so BRCA2, for example, in some set of mammals, it's in this small syntenic cluster, and in another set of mammals, it's in this larger syntenic cluster. And here's a few other examples. And so this paper goes on to, say that these genes that are in different syntenic clusters show us genes that were important for evolution of emergent traits, things that have to do with increased brain size. And I think there's probably some truth to that. And it's definitely interesting. But it's another thing that you can look at. You can ask questions about, for your gene of interest, is it syntenic with a bunch of other genes? Or is it one of these more rare genes that's in multiple syntenic clusters, which could imply more positive selection going on? Could. Plants, by the way, their histogram would not look like this. They actually have a very small number of genes that are in syntenic clusters across many different plant species. Side note. Okay, so that is all for this session. Hopefully you guys have quickly remembered what evolution was about, and you can go over positive, negative, neutral selection. Think about conservation, doing sequence alignments, multi-species alignments, how to calculate a conservation score, which has to do with the percent similarity per some background across different species, how you can use conservation to pick out pathogenic and benign variants. Ultra-conserved sequences are often markers of either coding regions or regulatory regions. And then this concept of synteny of things that are ordered in a similar way.